Regulation of fatty acid synthesis. Regulation of the genes involved in the synthesis of fatty acids such as acetyl-CoA carboxylase, ACC, fatty acid synthase, and other lipogenic enzymes is really critical to the lipid homeostasis in our cells. There's two key transcriptional factors that play really important roles for controlling these lipogenic genes. The first one is called sterol regulatory element binding protein 1C, also known as SREBP1C, which is primarily expressed inside our liver. SREBP1C is activated by a liver X receptor called the LXR receptor, which stimulates the expression of the SREBP1 gene. The activation will lead to increases in upregulation of lipogenic genes, and this will then cause the increase in synthesis of fatty acids and also triglycerides. Dietary polyunsaturated fatty acids, PUFAs, actually inhibit and turn off this gene. The second transcription factor involved in the regulation of lipogenic genes is going to be the carbohydrate response element binding protein, also known as ChREBP. ChREBP is activated in response to high carb intake or elevated glucose level. It plays a critical role in promoting the synthesis of fatty acids and triglycerides by upregulating the expression of these lipogenic genes. A glucagon activated cyclic AMP will inhibit this transcription factor, ChREBP, and also low energy via the AMP activated protein kinase, AMPK, also inhibits ChREBP. Insulin is known to stimulate fatty acid synthesis. However, on the other side, glucagon acts to inhibit this. So this balance of sugar intake and sugar depletion is really important for maintaining the balance of fatty acid synthesis and storage and proper lipid metabolism. On the right, you can see a picture showing you how this interaction works. On top, you can see PUFAs will inhibit the SREBP gene. Insulin will stimulate the LXR, as will stimulate this gene. Glucagon will then stimulate CAMP, and CAMP will actually inhibit the gene. On the bottom, you can see glucagon will stimulate the CAMP and also inhibit ChREBP. Low energy will also turn on AMPK, which will in turn inhibit the ChREBP. Glucose and eating a lot of sugars will activate the gene to turn on ACC, fatty acid synthase, and other lipogenic enzymes. Insulin and glucagon are two very important hormones involving the blood glucose levels and plays a really important role for regulating fatty acid synthesis inside our liver. Insulin is known to stimulate fatty acid synthesis, whereas glucagon acts to inhibit this process. Insulin promotes the synthesis of fatty acids by activating key enzymes involved in the pathway, whereas the glucagon opposes the action of insulin and begins to inhibit fatty acid synthesis. Glucagon's role is to exert an inhibitory effect on fatty acid synthesis by inducing the phosphorylation of acetyl-CoA carboxylase, ACC. This is activated by the CAMP-dependent protein kinase A, also known as PKA. Phosphorylation of ACC will then lead to its inactivation, therefore preventing the conversion of acetyl-CoA turning into malonyl-CoA, which was that key step we talked about. Insulin, on the other hand, counteracts this inhibitory effect of glucagon. It does so by activating a cyclic AMP degrading enzyme called phosphodiesterase 3B. And this degrading enzyme will then reduce the levels of CAMP, therefore reducing the levels of PKA, therefore preventing phosphorylation and the inactivation of ACC. So by the opposite approach, ACC is going to be turned on. Another important regulator of fatty acid metabolism is AMPK, AMP activated protein kinase. Glucagon will stimulate AMPK, which inhibits fatty acid synthesis, while insulin inhibits AMPK, allowing the activation of fatty acid synthesis. So just remember that glucagon is going to inhibit fatty acid synthesis, whereas insulin will activate fatty acid synthesis. Acetyl-CoA carboxylase actually comes in two forms. There is ACC1 form and ACC2 form. ACC1 is a cytoplasmic enzyme that's found inside the liver, mammary glands, and adipose tissue. It's primarily focused in the synthesis of fatty acids. Meanwhile, ACC2 enzyme is located predominantly inside the mitochondria and plays a role in fatty oxidation. ACC can be stimulated by citrate and inhibited by other abundance of acyl-CoA fatty acids. So as we said, the balance between these hormone signals and the activation status of ACC plays a very important role for regulating fatty acid synthesis. On the right, you can see a visual showing you that the citrate will then increase ACC, whereas other acyl-CoA fatty acids will then turn off ACC. ACC's main function is to convert acetyl-CoA to malonyl-CoA. Whenever AMPK is activated, or PKA is activated, these will then inhibit the ACC. AMP activated protein kinase, AMPK, is a critical enzyme that plays a central role in regulating energy metabolism in our cells. It phosphorylates not only acetyl-CoA carboxylase, ACC, but also several other enzymes inside the metabolic pathway. For fatty acid metabolism, ACC phosphorylates and inhibits ACC1. This will then lead to a decrease in fatty acid synthesis. When we inhibit ACC1, AMPK reduces the production of malonyl-CoA, and this will then result in a downregulation of fatty acid production. AMPK can also phosphorylate and activate ACC2. This will then stimulate beta-oxidation, which is that process we talked about for breaking down fatty acids. And the activation of ACC2 by AMPK will promote more entry of fatty acids into the mitochondria for oxidation. This will then help increase energy production and the utilization of stored fat. Let's talk a little bit about carbohydrate metabolism in this perspective. Carbohydrate metabolism also utilizes AMPK to play a role in glycolysis and gluconeogenesis. AMPK can stimulate glycolysis by boosting the enzyme phosphofructokinase, PFK, leading to an increase in levels of fructose 2,6-bisphosphate. When there's higher amounts of fructose 2,6-bisphosphate, this is the activator for glycolysis. It will then promote more sugar breakdown for energy. However, AMPK also can inhibit gluconeogenesis, the synthesis of glucose from non-carbohydrate sources, and by reducing the expression of the activity of the key gluconeogenic enzymes, such as G6P, glucose 6-phosphatase, it will inhibit gluconeogenesis, so AMPK does help lower blood sugar levels to maintain blood glucose homeostasis. Another key function that AMPK does in carbohydrate metabolism is its ability to stimulate the production of glucose transporter 4, GLUT4 receptors on the cell membrane. This will facilitate uptake of glucose into cells, promoting more sugar usage. So in summary, AMPK activated protein kinase phosphorylates not only acetyl-CoA carboxylase, but also other enzymes involved in fatty acid metabolism and even carbohydrate metabolism. AMPK can inhibit ACC1, thereby decreasing fatty acid synthesis, and activating ACC2 by promoting beta-oxidation. It can also stimulate glycolysis. It can also inhibit gluconeogenesis. It can also boost up glucose 4-mediated glucose transporters on cell membrane. One clinical application is metformin. Metformin is an anti-diabetic drug, which is considered a weak inhibitor for the respiratory complex 1 inside the mitochondria. This can lead to mild drops or reduction of cellular energy. The way this drug works is that it stimulates the glycolytic enzyme phosphofructokinase 1, PFK1, and inhibits the gluconeogenic enzyme fructose 1,6-bisphosphatase, thereby stimulating also more glucose 4 transporters inside the skeletal muscle, which is also alleviating more sugar out of the blood plasma. It also does target the AMPK activated protein kinase by raising the levels of that metabolite, fructose 2,6-bisphosphate. Let's talk about some diseases. Refsum disease is a rare genetic disorder characterized by a defective enzyme called the phenyl-CoA hydroxylase, PHYH. Phytanic acid is a branched-chain fatty acid that gets derived from the chlorophyll found in green plants and certain kinds of meats and protein. The methylated beta carbon of the phytanic acid does not get processed via beta oxidation. Instead, it goes through an alternative pathway called alpha oxidation, which is necessary to shorten that kind of phytanic acid. And it results in the production of propanyl-CoA instead of acetyl-CoA. So in disease states of Refsum disease, it is an inherited autosomal recessive disorder that affects individuals, and they have to have 2 copies of this defective gene. As a result of this, the disease manifests in the late childhood or even adolescence, and common clinical presentations include having weakness, visual impairment, retinitis, and even demyelinating neuropathies. And as a result of accumulation of phytanic acid, it can cause severity of the disease to range. The accumulation of phytanic acid in various tissues can cause the severity to be different and ranges from less severe to moderate to extreme severe cases. Another disease application is the Zellweger syndrome. The Zellweger syndrome is a genetic disorder that is characterized by a defective protein carrier of the peroxisome membrane protein, also known as MP70. This protein plays a critical role for the transport of saturated very long-chain fatty acids, ranging from 22 carbons to 26 carbons, into the peroxisome. Because of the absence or the malfunctioning of this PMP70 membrane protein, you're not able to transfer fatty acids into the peroxisome. And as a result, it accumulates inside the blood and tissues. This is a quite rare disease. It happens in 1 in every 25,000 males. And it does have multiple genes that code for this. So there are variations where there's 12 different genes involved in creating a certain form of the Zellweger disease. Some of the clinical presentations include difficulties with feeding, seizures, liver dysfunction, and also even high risk of death in early childhood.